Okay, as Monty Python said, for something completely different. So my name is Doug Doerfler. I'm the founder and CEO of Maxcyte. I was here, I think, 2 years ago, and we went public on AIM in 2016. What I want to talk about today is our company, obviously, but I also want to talk about a whole new area of science and a whole new area of medicine. And that area is cells, taking cells from patients, re-engineering them, and giving them back to patients for treating diseases. And Maxcyte's in the center of this new area. And what I want to try to get across today is an interest that you will have in trying to learn more about cellular therapy, using cells as a way of treating patients for diseases that really can't be treated today. So we are based in Gaithersburg, Maryland, in the US. We went public in 2016 on AIM. We raised about 30 million pounds since we went public. 10 million on our IPO and 20 million in a secondary that we did in 2017. And we sell product, we license our technology, and we also develop our own therapeutics, and everything's based on our one technology. And it's really for creating drugs that are your own cells, taking your own cells and turning them into treatments. And we're involved in the treatment of cancer, infectious diseases, genetic and inherited diseases, a broad range of diseases, and cells are now a new class of drugs for treating these diseases. We also have our own product. It's called Karma, and Karma is a very rapid way of taking cells from a patient, engineering those cells, immune cells, so they recognize cancer is foreign and kill cancer cells. We have submitted our IND, which is a request by the Food and Drug Administration, which allows us to give this drug to other people. And we expect to be treating patients this year in 2018. We also license our technology. We have over 50 programs where we license our technology to companies who use our technology to make drugs with cells. And I'll talk a lot about this, about the portfolio of products that we have. And we're involved in a lot of really cutting-edge science, which I'll talk about. But underlying all this whiz-bang science, we've got a growing business. We grow the business about over 10, 20% a year, top line, double-digit growth. Gross margins are 90% now. And we currently have, well, at the end of 2017, we had $25 million in revenue, in cash in the bank. So let me talk a little bit about what we do. So we have a basic technology, and this basic technology uses electrical forces to put molecules into cells, blood cells. So you take a bag of cells from a patient, you run them through our system, and we use electric forces to drive a molecule into the cell that's been specifically designed to engineer that cell to do something different, maybe to grow into a different cell, to attack an HIV virus, to attack a cancer cell, which it normally would think it's its own. And we use this technology in a number of different areas. But what's important about what we do is that we are able to take cells from patients, very rapidly engineer those cells to give back to the patient. We do it very efficiently. We do it without any contamination. And we do it in large volumes, so you can get enough cells to actually treat a patient. And that's what Maxcyte's all about. And that's our proprietary technology. So everything we do is based on this one technology. We invented the technology. We licensed the original technology from the Center for Blood Research at Harvard. We put together a group of physicists and biophysicists and blood experts to create this technology that allows us to modify cells. We have about 50 employees, over a third of whom have either PhDs or MDs, so it's a very heavy science business. And we also have people around the world who are our employees who license our technology to the major pharma companies and biopharma companies. We really do it in 3 areas. The biggest one is Karma, which is our wholly owned proprietary platform that we invented. I'm going to spend a bit of time talking about this for treating first ovarian cancer and mesothelioma, 2 very, very difficult to treat diseases. Secondly, we partner the technology to companies to then use our technology to make drugs and take cells and turn them into drugs. And we have over 50 partner programs. Over 20 of those are in the clinic actually treating patients. And these clinical trials include using cells, so your immune system, the reason cancer grows unchecked is because your immune system doesn't recognize it as foreign. Now, contrast that to if you walk into a room and there's an allergen and you get a sniffle, and it happens just like that. Well, cancer is very dangerous because what it does is it evades your immune system and it goes unchecked. And your immune cells are probably the most powerful drug that you can ever imagine. But the cancer cells evade the immune system. And what we do and other companies do is re-engineer your immune cells so it recognizes those cells as foreign and then attacks it and kills it. So we're working in that area. We're working in the area of gene editing. This is where we can take a stem cell from a patient and edit the genes and give it back to the patient. It's called CRISPR technology. It's one of the ones many of you may have heard about. It's been on the front page of The Economist, the Financial Times, a really new revolutionary way of editing cells. And Maxcyte's in the center of that. We're also working in regenerative medicine. This is regenerating liver tissue, regenerating kidneys. And again, our role in this is an enablement. So we're not the company developing the products, we're the company that makes the products better. That's the old Intel Inside or BASF. That's our business, is being able to provide a very, very specific technology that can be used in the design, development, and delivery of drugs. We also sell our instruments to big pharma. These are Pfizer, Novartis. 9 of the top 10 pharmas are our customers. They use it to discover small molecules. They use it to produce proteins like monoclonal antibodies, something called erythropoietin. Again, biological drugs that many of you may be familiar with, or your families may be familiar with. Let me talk first about CARMA. As I mentioned, CARMA is in with FDA. We expect clearance this year, begin to treat patients this year. This is a really new foray for us, and it's a very new, exciting class of drugs. And this is really based on leveraging experience we've had in this field for close to 20 years. So we're not kids. We've been working in this field for a long time. We understand it. And that group that actually invented the technology I talked about is still in the technology, still within the company, working and trying to solve these problems that our customers are trying to solve to create better drugs for patients. So I'm going to talk a little bit about T-cell immunotherapy. And you may not have heard of this, but if there's one thing that I ask you to please do tonight or tomorrow, is to Google this area. It's called CAR-T. It is one of the most fascinating areas of science there is right now. And there's 2 drugs that have been approved. And these drugs really came into the forefront in 2014, 2015. And this is where they take patients' cells, their white cells. In this class, they're T cells. And T cells are very efficient at killing things that it determines are foreign. And what these companies are doing is they're taking those cells and using a virus to engineer the cell so it turns against the cancer cell. And they've had phenomenal results. They've had close to 400 patients that have been treated in clinical trials. These patients have had 5, 6, 7 courses of therapies. So these are end-stage patients. 1/3 of these patients have walked out of the hospital in complete remission. Now, this is a very highly controlled scientific study. It's been well published with all the journals. And what happened, these 2 drugs were approved. One is Chimera, which is a drug that is being developed by Novartis. A second drug is called Yescarta, which is being developed by a company called Kite Pharmaceuticals. And then there's a third drug, a third CAR-T drug. Again, all 3 of these are against the same target, is by Juno. Now, Juno was acquired at the beginning of this year for $9 billion by Celgene. And at the end of 2017, Gilead acquired Kite for $12 billion. So here are drugs that have, these companies were startups 4 years ago. And over the course of those 4 years, they've been acquired for $20 billion because of the success of these T-cell-based therapies. Now, as I mentioned to you, we have been involved in this field for a long time, and there's a lot of big pharma companies who have jumped in and have done deals with these development stage companies. So these are the big pharma companies, and these are the companies like Maxcyte that are developing the drugs with the academic centers. When we talk to these companies, there's a handful of big pharma companies that are missing. And there are companies like Lilly, there are companies like Roche. And we talked to them about why you haven't jumped into this field, and they said really three things. One is that everyone is focused on blood cancers. These drugs are all leukemias, for instance. Leukemias represent 10% of the oncology market. The whole oncology market is $100 billion a year in revenue, and 10% is blood cancers, 90% is solid tumors. These drugs don't work in solid tumors. The second thing is, it's a very expensive and laborious manufacturing process. And those are the two other reasons. It takes about 4 to 5 weeks to produce this product. So you go into the hospital, you get your cells taken, you do the genetic manipulation using a virus. After 4 or 5 weeks, the doses are ready, you go back in and get that treatment. Now the problem with that is a couple. One is that it's very difficult from the patient's perspective. Just think about the fact if you went in and got a prescription for a drug, but you had to wait 5 or 6 weeks to actually get the therapy, that's a problem. It's a different situation if you're on your last call, but if you're not, it's a very difficult situation for the patient and their family. Secondly, it's extraordinarily expensive. The selling price of these drugs are between $400,000 and $500,000 for a single course of treatment. And so you can think about this maybe in some major countries like the UK, but you're not going to find it in many of the other countries because they just can't afford to, their health systems just can't afford to cope with that. So we have been working in this field for close to 10 years, and we've been working with the top people in the field. And the top people in the field, the number one guy is a guy named Carl June at University of Pennsylvania. We've been working with him. And a gentleman named Dario Campana at the National University of Singapore. That white block that we couldn't figure out, that logo is actually the National Institutes of Health in Bethesda, Maryland, which we also work with. And we're working with these groups because what they were concerned about is they wanted to be able to use this kind of an approach in solid tumors. And I mentioned before that these CAR Ts are using viruses, and a virus will affect the cell and engineer the cell, and that cell is permanently engineered. So when you give that cell back to the patient, it's always surveilling for a particular protein on a cell, and if it finds that protein, it attaches to it and kills the cell. Well, there's very few cancers, solid tumors, where that little protein called an antigen on the cell isn't also found on normal tissue. And that's where you have very high chance of very significant toxicity that can lead to death. And so what they were working with us on, our system uses electricity, not virus. So we can put a molecule into the cell and engineer that cell for a short period of time. So instead of it being forever engineered, it's engineered for 5 or 6 days. And then it goes back to its normal state. And that allows us to go after solid tumors. And that's our approach. We've been working with these companies, these universities, to make this happen. But what we did was we figured out something different. We figured out with our technology, we can do this very rapidly. We don't have to do this 4 or 5 weeks. We can do this process in a matter of days because our technology, again, uses electricity. It's very rapid. We can do it in large volume. So what we have is we have begun our preclinical work at Karma, and this is the cartoon for how this technology works. So you take white cells from a patient, so this is called apheresis. Their apheresis instruments are found in virtually every hospital in almost all oncology centers. And what it does, it takes your blood and it separates out your white cells. And again, your white cells are your immune cells. You take those cells from the patient, that's a 100 milliliter blood bag, it's about this big. You add in what we call messenger RNA, which is a molecule that expresses a protein but only expresses for a short period of time. We use our technology, which is this reddish instrument, that's our system, puts the molecule into the cell, and we can do this entire process in less than a day. So it dramatically reduces the cost, it dramatically reduces the complexity, and we think it's going to dramatically reduce the cost and therefore the selling price to patients. And we'll be able to go after solid tumors, and we will also be able to move forward very rapidly into the next stage of clinical development. So, little science here, we're going after an antigen. So the antigen's called Mesothelin, and I mentioned that this is a protein that's found on cancer cells. Now, some of you may have had a PSA test. PSA means prostate serum antigen, and so they're testing for that. They're testing for an antigen. So this is a very well understood area of identifying antigens. There also happens to be an antigen called mesothelin that is found in a number of different cancers, which include ovarian cancer, mesothelioma, bile duct cancer, esophageal cancer, colorectal cancer. And these are all potential targets for our first product, which is a mesothelin directed CAR product that's using fresh cells. That's what Karma is all about. So I'm going to skip over. So if you have any questions, I think we'll wait to the end. But that's the Karma program. The other thing that is driving revenue for the company is in partnering this technology with cell therapy companies. This isn't an eye test, but what we're doing is we license our technology to these companies for them to do work, and they pay us during the process. They take all the risk, and we receive revenues for it. What we're also finding is that these are some of our partners, and in the field, these are some of the top companies in developing cellular therapies. And they're developing these programs in a number of oncology applications. And in that center block, those indications are those that are currently in the clinic treating patients using our technology. During this process, so our business is really to build out this enablement, license the technology as broadly as we can with companies who are paying us for that early stage in clinical work, and eventually when they move to commercialization, they'll be using our technology to actually treat patients on a commercial basis. This area is growing significantly. We went public. We had about 30 to 40. Now we have well over 50 partnerships in place with some of the leading companies. And one of the reasons why this area is so hot is this bottom left-hand box. In 2017, there was over $7 billion invested in this space around the world. $7 billion in fresh capital going into this space, and $2.2 billion in corporate M&A payments to these companies. Now, this obviously doesn't include the $20 billion that was invested in the two companies I talked about before. So this is a very, very important new area of science, and one that many of us and we believe is going to be really the future, and be able to solve a number of the intractable problems that we have as a population. Our business model is, again, to get as many shots on goal as we can. So we have over 50 programs in the research. Now, when you're in research, we charge our customers $150,000 per instrument per year. We don't move the title to the instrument to them. We retain title to the instrument, but they pay us a licensing fee, $150,000 per instrument per year. When they move to the clinic, the price goes up to $250,000 per instrument per year. Typically, they get multiple instruments, so these deals start to really build. Eventually, those two blue circles denote a commercial deal. So this is where a company called CRISPR Therapeutics, who's using our technology to edit genes. They're editing genes for two indications. One is for sickle cell disease, which is a deadly disease, and the other one is severe combined immunodeficiency disease. This is the Bubble Boy. Have you ever seen that movie, The Bubble Boy? This is where those patients have no immune system at all. And what they're doing is they're taking the stem cells from these patients who can't fight any diseases, they can't fight any bacteria, and reprogramming their immune cells so their immune cells can actually fight these diseases so they don't have to live into a bubble. And so this company paid us $1 million up front after they have been paying us for research and development. And then we have milestones and then royalties that are behind that as we work with these companies. We have 2 of these deals signed and announced. Our business really is to build the research part of this, help our companies move to the clinic, and eventually commercialization, where we think there's a tremendous upside for this business. So this is a part of the, I think this is part of the reason why we were able to get the investors in with our offering, and we see that the share prices has changed because we're making great progress in this part of the business. Our investors include BlackRock, Legal & General, River Mercantile, so we've been able to put together a really nice investor group for this, who I think understand the future of this. It's a bit complicated, but if you think about number of shots on goal, working with these companies in some of these diseases that can't be treated otherwise, and as they move to the commercial sector, we're going to be part of that approval process, and we're going to be able to support them and sell instruments and disposables as they're treating patients. So every time a patient's treated, they use a single-use disposable. So this is the razor blade model. But in our case, the razor and the razor blade both have 90% gross margins. So we think it's a pretty attractive business. The third part is where we actually sell the instruments. We sell them to big pharma companies. We have close to over 200 instruments now in the field that are being used by, again, Novartis, the top companies in the field. And they're using it to find new small molecules. We work with AstraZeneca. They use our technology to find new drugs for psychiatric disorders. A company called Vertex for cystic fibrosis. So there's a lot of different areas that we're working in. But what we do is focus on putting molecules in the cell. That's what we're really good at doing. And our customers rely upon us to provide that support. 12-month news flow. We have our IND cleared. We expect to be dosing patients this year. This is a big deal for the company. This moves us into a whole new area of development as a company, and expanding our CARMA programs. Additional cell therapy programs we'll be moving toward. Expanding the leadership team. We just brought on a senior person as a director, corporate director, Richard Douglas from Genzyme, a company you may have heard of. We just hired in a new chief medical officer from Takeda Pharmaceuticals who just joined us. So in summary, we will be the first in human this year with a new drug called CARMA, which is a fresh cell to treat ovarian cancer and mesothelioma. Focus on driving the adoption of this technology across additional cell therapy companies, helping them move their programs into the clinic and eventually commercialization. And then building out a instrument sales and licensing business. Again, double-digit growth, 90% gross margin. So this is a bit of a hybrid. You've got the defensive part of this, which is a system that we're selling instruments and single-use disposables. And then the upside is using the technology to create new drugs for our partners and eventually our own proprietary drug platform called Pharma. Again, revenues, we did $14 million last year, was about a 14% increase. We've had a mid-20s over the last several years. We invested $7.5 million in CARMA. That'll be slightly higher this year. We run the base business at a slight operating loss, about $1.5 to $1 million in loss. We do that. Our board's very comfortable with that because we see this field just exploding. And we think it's a land grab, and we want to make sure that we're out there working as closely as we can with our customers, solving problems for them before they actually figure out they need to solve the problem. So let me just stop there and thank you for your attention. Didn't put very many of you to sleep. And if you have any questions, please.
Time for questions. Anybody got any questions? I know there'll be some on the front row here. Oh, there's one.
Yes,
Not
sir.
the person I thought.
Yeah, electroporation has been around for decades and decades.
Yeah.
So in what way is your technique different from what's been around for years?
Yeah, so it's been around probably since the early 80s, and it was used originally for putting molecules into bacteria, not mammalian cells of bacteria. We're the first really to focus on mammalian cells. So mammalian cells are what we have in our body. Bacteria are different. And we were able to do it in much higher performance than you typically would see in electroporation. We can do it completely sterile. We can do it in large volume, and we can do it clinically. So we're completely differentiated from that technology. But we use the learnings of that to create this new instrument and this new technology.
So you have IP for all that?
Absolutely. We're very well protected in intellectual property. So yeah, good question.
Next question.
Doug, thank you. I would freely admit to being confused about the the total package that you guys are offering, but I'm particularly interested at a personal level in your ability to edit stem cells.
Mm-hmm.
So you are actually thinking of taking autologous stem cells, which is stem cells from the person, editing them in some way and putting them back into the patient to cure various diseases. Can you explain that a bit more?
Yeah, we're not thinking about that. We're doing that. The field's doing that. So we have, I mentioned the sickle cell disease with CRISPR therapeutics. I mentioned the bubble boy disease. They're actually doing that, and they're about to, and I think they've gone into clinical trials with the first one.
But these are your technologies, or
Yeah.
these are...
So our technology delivers the molecules into the cell. What we do is we deliver molecules into the cell. Our partners design the molecules. That's what they do. We provide them the means for the delivery. We're working with NIH, for instance, also in 2 diseases. We just recently announced the second one a couple weeks ago. Again, it's almost like science fiction. It's correcting a single nucleotide. So these are diseases that one nucleotide, one piece of DNA different from normal to diseased. Find that piece of DNA, knock it out, and replace it with a corrected version. And that's the new field. People are working in this area across the board in a number of inherited genetic diseases. Very, very interesting area. What we want to do is we don't want to take the risk, frankly, in that, because each of these companies have set up their own, raised hundreds of millions of dollars to do that. What we want to do is provide them the enablement and the technology for them to do that kind of work.
Doug, in your Karma study, you're targeting mesothelin.
Yes.
What side effects would you predict, if any, from that treatment?
So we think mesothelin is a great target for us to prove something that no one's ever been able to prove before, and that is that the problem with treating solid tumors is you've got an antigen on that solid tumor that's also found on normal tissue. And so if you use a virus-based CAR, that never stops. It can't differentiate between a normal cell and a cancer cell. Our technology, our Karma drug, only lasts for 5 or 7 days, and then it goes away. And so we believe that we're going to be able to differentially attack cancer cells and leave the normal cells alone, because cancer cells do express that antigen at a much higher density than normal cells do. I don't know why that occurs, but it occurs, and we're going to use that phenomenon as a way for us to be able to give a preferential killing to tumor cells versus normal tissue.
Okay. And how often do you anticipate dosing the patients in the Karma study, and for how long?
Yeah, so we haven't disclosed the clinical trial design at this point, but we expect, based on the preclinical work that we have published with Johns Hopkins, we did a lot of work with Johns Hopkins University in Baltimore, we'll be able to dose the patients every 1 or 2 weeks, and a single apheresis will give us somewhere between 3 and 6 doses. So you'll come in and you'll get your cells taken, processed, you'll come back every other week for, let's say, 4, 8, 10 weeks, getting your treatment, then you'll come back and get another apheresis and get more treatments until you can show a remission in the cancer or elimination of the cancer.
And is there any way of arresting the degradation or the reversion of the cells back to where they were if you freeze, or?
So when we actually process the cells, we freeze them all.
Okay.
So when the patient comes in, they're only getting the cells. They're not having to give an apheresis. They're just getting a dose of cells every other week. The apheresis will come in maybe every 2 or 3 months.
Okay.
So it's frozen.
And can I ask about the CRISPR therapeutic study? Are you using, you're doing that with stem cells, so is that from apheresis fluid?
Yes.
It is. It's not.
Well, they'll take apheresis. So stem cells are typically in your bone marrow,
Yeah.
and they can either take them from your bone marrow, or they can actually take them and separate them from your peripheral blood.
Okay.
It's a very rare event in your peripheral blood, maybe less than 1% of the cells, so it's really like a needle in a haystack trying to pull those out. Typically, they'll do a bone marrow pelvic tap to get the stem cells. Bone marrow is rich in stem cells, and if you have one of these diseases, that's something that you're going to probably do,
Yeah.
because it's such a debilitating disease that the therapy is worth the discomfort to the patient.
Okay. Thank
Final
you.
question at the back. I'm going to have to stop you there. Final question.
Can I quickly? Can I just get you to explain perhaps the proposed business model for the Karma? I mean, I know it's some time away, but I mean, do you envisage working with pharma companies to do this, or would you envisage that this, effectively the cancer centres might actually be the commercial partner for you, for example?
It's a great question. We are not going to be a drug developer. That's really not what we want to do. So we know what our technology can do, and we're building out clinical trial groups that we can do phase 1, phase 2, and we actively have a business development group. The guy who we brought in on our board of directors from Genzyme was head of corporate and business development for Genzyme for over 30 years. So we're going to be working with pharmaceutical companies who have different targets. We've selected mesothelin, but there are scores of other targets that companies have and have studied, and we think we'll be able to partner with them to use our platform, which again is that rapid way of turning a cell into an anti-cancer with their target. And so that will include us getting co-development rights, and them licensing fees, because again, we've licensed over 50 programs. So we're a technology development and licensing business, and that's what we're going to continue to do. What we want to do is invest in clinical data, because that's what pharmaceutical companies want to see. They like to see stuff in mice, but they would rather see stuff in humans, and that's what we're doing, investing the money to show that this actually works in other people.
If I might just follow up with one, how do you envisage that it will be regulated? I mean, what's the regulator's position on these cell therapies? Is it any different to a pharmaceutical?
It's different because the product is actually the process that you use to develop the cells, so it's product by process. But there's a center for cell and gene therapy at FDA. And MHRC here has a wonderful group that is very collaborative, working in the cell therapy area. So the regulators have become very comfortable with regulating this sort of therapy, but they want to make sure that everything's done under CGMP. They want to make sure it's sterile, and that's where we come in, because we have a sterile, closed system, and we have regulatory authority in the UK, Italy, Germany, the US, Canada, Japan, Thailand. That's what we do. We work with these groups to ensure that they can use our technology in their CGMP manufacturing processes.
Doug, thank you very much indeed.
My pleasure.
Thank you.